After that, they get these two and put it into different samples. So there is one sample with artificial seawater as the environment. So what is this artificial seawater is that they added their own mercury, they added zinc, they added copper, and they added cadmium into this artificial seawater. So one of it will have the biosensor and the other one will have the fish in there. And they also have another type of environment, which is seawater that is collected from the field itself. And the same thing, they put the biosensor in there, and another one, they put the fish in there. And after that, they analyze this with Pearson correlation and also with ICP-MS. So from the results and discussion, we can see that it shows significant correlation. So that means that the performance of the biosensor is comparable to the fish. And also through ICP-MS, they detected significant correlation. So as you can see from the graphs, when the concentration of the heavy metal increases, the luminescence inhibition ratio also increases. Alright? So this is what we understand so far from the explanation from the beginning of my presentation until now. Like when the concentration of heavy metal increases, it will affect the cell, it will affect the reactions in the cell, it will affect the bacteria, and then some reactions will be inhibited, and thus we can plot the graph of inhibition ratio versus the concentration of heavy metal. So in conclusion, they concluded that when the cells of TOX2, the bacteria, are exposed to toxic compounds, the machinery of the gene transcription and protein translation in the cell will generally be inhibited due to the cytotoxicity, and thus this will lower the intensity of the bioluminescence of the bacteria. So when the bioluminescence is reduced, the signal produced will also be reduced. Or if you were to calculate it using inhibition ratio, the inhibition ratio will increase. So these are the references that I use for my presentation, and that is all for my part. Now I will pass the presentation to my friend Arshad, who will continue with the presentation. Thank you very much, Doctor, and thank you very much, my fellow friends.